I'm Doug Lauffenburger, an old friend on this side of town, and Linda Griffith are both members of the Koch Institute and leaders in the field of bioengineering. And I think Dr. Lauffenburger is going to talk to us about his and Dr. Griffith's work, who was unable to be here this evening.
Right, that depends, because then that would take longer, so.
Well, you got 25 minutes.
Do I have, I get her time? Wow, wait till I tell her that. So, no technology here, no tools. One thing that sometimes flies under the radar is that folks with an engineering background can just bring a different way of thinking rather than tools, devices, and so forth. So that's really what I'll show you in the next few minutes here. So what we're after is, if you have a complex system like a tumor, and especially a tumor microenvironment, so just to give away the game, what this is about is trying to do what folks call systems biology, which is just really dealing with multiple variables at once, but to do it in vivo, to do it actually in living systems, animal models or humans, because you can't capture all these things in cell culture. And the goal of it is, can you learn enough, can you put enough of it into some kind of a model that's both conceptual and computational, that you can actually predict what's going to happen when you intervene with a putative drug, or what happens if there's a mutation and so forth. So this may give you some idea of what would happen then when you intervene with targets, even combinatorial you can do this way, whether they're intracellular small molecule pathway inhibitors, cytokines, chemokines, even cellular depletions. And here's the important notion at the bottom, and that is actually lots of things matter. There's no magic bullet target. There's no single best target. That in complex diseases, there's lots of targets. There's lots of ways to treat it. There's lots of ways to drug it. The problem is not a dearth of targets. The problem is a lack of ability to predict what happens when you actually drug them. Okay. So that's really what we're after, is to say, out of the multitude of things that are all going on in these complex systems, microenvironment and some pathology, can you really put together understanding of what happens when you perturb any of those moving parts so that lots of things happen dynamically, and can you in fact understand the outcome? That would then allow you to choose among multiple different types of putative targets, all of which are there, because then you actually have some hope of predicting what's going to happen. So here's the thought process, and this is just paradigmatic engineering. You have a complicated system. In this case, all that's shown is one cell. So this would be one cell with signaling pathways that govern transcription and metabolism and cytoskeleton that then govern phenotypic responses of proliferation, migration, death, and so forth. And so the idea is to really capture a robust model for this, you just need to measure a lot of the key aspects. So in any given cell type, you need to measure multiple molecular pathways or effectors. You need to measure if there's multiple cell types in a tissue. You need to measure the multiple cell types. Associated phenotypic responses, so you can see how these regulatory activities may relate to them. Most crucially, you have to undertake this across a broad landscape of conditions. If you have any hope of capturing what's happening physiologically, there's no single experiment that you're going to do that's going to capture that. What you have to do is many different experiments in many different environments, and the computational model integrates the behavior across all those environments and allows it to be robust and allows you to predict what's going to happen outside of any particular experiment. Otherwise, you can't predict anything other than your given experimental conditions. And of course, you can't do this by your own intuition. Put together these numbers of measurements, components, conditions. Only computational analysis will allow you to do that. So with Kevin Haggis, who's at the Cancer Center at MGH Charleston, we've demonstrated this in mouse models of a number of diseases. I'm going to show you something very simple here. And it may seem like it has nothing to do with anything you're interested in, because all it's going to be is tumor necrosis factor-induced intestinal inflammation. What happens to intestinal epithelial cells? Do you dysregulate apoptosis and proliferation in response to things like TNF? I think if you envision it properly, you can imagine this as a tumor tissue in a microenvironment in response to drug perturbations. The paradigm is exactly the same. The methodology is exactly the same. The thought process is exactly the same. So you can think of it, you have multiple mice with multiple treatments. You might have different genomic backgrounds. All of that's fair game to put together depending on the scope of the model you have in mind. You have some kind of stimulus or perturbation. In this case, it's just going to be different doses of tumor necrosis factor. You can imagine different doses of drugs of different types. And then you have to measure things in the tissue. And we're going to measure a lot of things. We're going to measure signaling pathways inside the cells. We're going to measure the cell types that are there. We're going to measure extracellular cytokines, chemokines, proteases, and so forth. We're going to measure the phenotypic responses. And then the computational modeling is going to pull it all together and tell us how the moving parts interact and affect each other and what happens if you perturb any of them. So just to run through the types of data, and again, you could imagine this in tumor tissue biopsy and microenvironment if you'd like. So you could measure the behavior of the key cells, tumor cells. In this case, it's epithelial cell proliferation and apoptotic death under various different treatment conditions, TNF concentrations, different genetic backgrounds, RAG1 knockouts, RAS mutants, lots of things. In the epithelial cells, you can then measure, along with their phenotypic behavior, activities of the key pathways. And you can think of all the pathways you'd like to imagine, the different kinase pathways and so forth. You can measure them under all these conditions. You can measure what's going on in their microenvironment, the cytokines, the chemokines, the growth factors, proteases. You can measure all of those under all those various conditions. You can measure the cell types that are there by flow cytometry, what's happening with the immune response, what's coming in and going out, innate, adaptive, stromal cells, under all these conditions. And so what you've done is essentially you've pushed and pulled this tissue around with various perturbations, and the cell types change, and the cytokines and chemokines and growth factors change, and the epithelial cell signaling changes, and their phenotypic response changes across a lot of different conditions. And then you need computational analysis that say, how do all these threads pull together? What cells are affecting what molecules are affecting what cells, what signaling pathways are thereby affected, and then what are they affecting, and so forth. Okay, and we can do this, and if you look at the publications, we can do this. So just to show you the kind of thing that comes out is, in a simple situation like this, we can pull out essentially the logic of interactions of multiple cell types. In the end, what will cause intestinal epithelial cells to become hyperapoptotic to TNF-alpha? The TNF-alpha itself doesn't do that much. What happens, they get sensitized by interferon gamma. Where does that come from? It comes from plasmacytoid dendritic cells that are normally not there. Why are they not there? Because goblet cells are making MCP1 or CCL2. What's governing that? Cytokines that the T cells are making. Okay, and you can pull together these threads, and you can pull together them quantitatively and computationally, so I can tell you if we deplete MCP1, if we deplete plasmacytoid dendritic cells, we deplete T cells, so forth, add molecules back, add kinase inhibitors, we can tell you what's going to happen to the system. Okay, and this is in vivo data, and this is multiple cell types in the microenvironment. To show you that we know what's going on biologically, all of these manipulations, everything that happens in the microenvironment, whether you change cytokines and chemokines, whether you deplete immune system cell types, whether you add cytokines back, and so forth, if we understand what's going on, everything should affect, if we're interested in the intestinal epithelial cells and whether they proliferate or apoptose, it should affect the signaling pathways in the intestinal epithelial cells that govern those responses. And if we measure those signaling pathways that we do, there should be a single model that says you add up those pathways under all the different conditions, and when you add them up in the right way, that it'll tell you that no matter what you did to perturb the microenvironment, you can predict whether you've gotten more apoptosis, proliferation, or less. And that's all demonstrated here, that there's essentially an eigenvector, a way to add up multiple kinase pathways inside the epithelial cells. And when you add them up, under normal conditions, not much happens, and the cells are relatively benignly affected. Deplete MCP1, it says this is what happens to the intestinal epithelial signaling pathways. Add them up, it says, aha, Now I've got pathology. My epithelial cells are crashing and burning. That's what happens. If I had RAG1 knockouts, that changed that whole cause of events of MCP1 and goblet cells and plasmacytoid dendritic cells and interferon gamma. That changes the signaling pathways inside the epithelial cells in a way that exacerbates the disease. If I just deplete the plasmacytoid dendritic cells, a lot of those things unravel, and in the end, if I just measure the signaling pathways inside the epithelial cells, then it tells me they're fine again. So what it means is there's mechanism here. There's actually biological insight here. This isn't just some sort of numerical abstractness. It's all boiling down to signaling pathways in the cells you care about, governing their phenotype, all of which is modulated by everything you've done to modify and alter and perturb the microenvironment. Okay, so I can really imagine trying to do this in whether you want to do xenografts or genetic models of tumors and so forth. There's no reason we can't do that. Okay, the other part, as long as I get some of Linda's time, this is a project we've done together. And the idea here is, what can you take into human patients, not just mouse models? And this happens to be with a disease called endometriosis, which is an invasive disease of ectopic cells being someplace they don't belong. And in patients with the disease, instead of being cleared by the immune system, they're allowed to persist and invade, and they form lesions. So this just happens in the peritoneal cavity, that endometrial epithelial cells actually can penetrate tissues and survive against the onslaught of the inflammatory and immune system, form lesions that are debilitating. These aren't exactly malignant tumors. Nonetheless, they're invasive lesions that have formed in places that they shouldn't. So can we take a similar approach? The concept is the same. Although here what we decided is we'd work backwards, because from human patients, you can't do all those perturbations to construct the model, right? So can we work backwards and, in fact, do enough perturbations and conditions in cell culture, and then from that get the insights that project back into the human patients? So you might think of this as the inverse. So the approach was the same, but just starting in cell culture with the invasive endometriotic cells. What makes them invasive? Proteolytic activities, breaking down the matrix, migrating in, stimulated by growth factors. What's governing all of this? Proteolytic shedases at the cell surface govern what growth factors are produced, govern what matrix metalloproteases are activated. So we've got to measure all those things that are activated at the cell surface. We've got to measure the proteolytic activities. What are they governed by? They're governed by kinase signaling pathways. Okay, so we've got to measure all of those. In the end, what's the phenotype that we care about here? It's invasive migration, so we measure that too. So same thing, under all the different types of growth factor and inflammatory cytokine conditions we can think of, these cells in culture, we measure all of these different things. Ends up looking something like this. We're measuring phosphoprotein signaling under lots of conditions, protease activities under these conditions, ligand receptor shedding in the environment, the migration. I will take 1 minute and say something about a tool here, and that's the protease activities. That's the hardest thing to measure because there's no such thing as a selective protease substrate. Okay, if anybody tells you they've got a protease substrate that'll tell you what's going on with atom 10, it's just not true. None of these are specific. What one has to do is a whole different technology we've created that we call PRAMA, for proteolytic activity matrix analysis, by which you can use recombinant protease substrates across a whole battery of proteases and get activity signatures that then you can go back and deconvolute to the measurements that you actually make with whatever substrates you have in your biological sample. So you can't depend on a protease substrate. It's just not specific enough. You have to do this battery. All right, now you pull these data together, and you can build essentially a dynamic network model that says what's upstream and downstream and what's the logic by which growth factors and cytokines govern the kinase pathways. How do they crosstalk? How do they affect protease activities? What substrates are those proteases acting on? And once you think about the activities the proteases are acting on, some of them are matrix proteases that then degrade the matrix and lead to migration. Some of them are shed growth factors that come back and form positive feedback loops. enhancing the activity. Some of them are negative feedback loops because you're shedding receptors. And if you shed a receptor, that's actually a negative feedback loop because the ligand might be there, but now the receptor's gone. So all of this is happening dynamically in your cells. So you need a dynamic model like this to say, okay, whatever environment the cells are in, if I perturb a kinase pathway here, it's not as if just that pathway is perturbed. This whole dynamical positive and negative feedback loop is perturbed, and the outcome of that is not obvious unless you do the computation. So what are the consequences of this? What it means is you can sort out why some kinase inhibitors work and why other kinase inhibitors don't work, because along with inhibiting a pathway that you think governs migration, they're also affecting the proteolytic activities, affecting the positive and negative feedback loops. And so some of the kinase inhibitors will inhibit migration under some conditions, but actually make it worse under other conditions. Okay? And so you have to figure out what combinations you would need. Now, that's all cell culture. How can we actually get it to the patients? So the proposition is, if it's these activities that are driving these endometriotic cells to invade into the peritoneal tissue, then we ought to see the signatures of these feedback loops in the peritoneal fluid that's then being modified by these cells as they're forming these lesions. So we did go into endometriosis patients with Keith Isaacson and Newton Wellesley Hospital and measured all these things we could find in the environment. I guess this is another technology. It's a nanodroplet multiplex protease activity assay using the same kind of deep convolution I told you before, because even with fancy nanodroplets, the protease substrates still aren't selective. So you can measure all these substrates under these different conditions. You can measure all the activities, therefore, of the Adamses and NMPs and so forth, and compare them between disease patients, endometriosis patients, and controls, and see if this positive and negative feedback loop is represented in diseased versus normal patients. Interestingly, it is. So if you look at the patients that we have in the middle in this blue, this is projected into a principal component space, just a multivariate analysis way of really finding the key combinations of all these molecular components. These then are the normal patients that we would then conclude have the right balance between the positive and negative feedback loops, and you're not getting the invasive migration. Interestingly, there's a group of patients over here that have all the signs of the activities of the dysregulated positive feedback loop driving migration. And then there's another group of patients in which the feedback loop is dysregulated in the opposite direction. And so we actually believe there's two forms of the disease, one that's driven by an overactivity on one side of the feedback circuits, one that's driven by an overactivity of the other side. To treat them with the same drug would give you, even though it looks like the same invasive disease, would give you two different types of outcomes. And so you have to be able to recognize them like this. So in endometriosis and some other type of now inflammatory invasive diseases, we think we're being able to bring this kind of approach actually into human patient samples. Different than mice, of course. So you've got to go about it a little bit differently, but the paradigm's the same. So.
Thank you, Doug. Questions? Please.
Can you distinguish ovarian cancer from the acidic fluid in endometriosis patient?
Great question. Was about can we distinguish ovarian cancer? As you know, Jim, I was just up at Ontario Cancer Institute visiting Ben Neal, who's really digging into ovarian cancer now, gave exactly this seminar, and we're starting to work together on exactly that he thinks by this approach we're going to be able to do something to figure out actually even some of the types of ovarian cancer. Yeah.
Question? Please, Jerry.
How many parameters do you need to measure to get a robust model?
Yeah, how many parameters do you need? It's not so much number, it's what they are. So you have to know enough about the biology to say what pathways, what cytokines and chemokines, what proteases. And so in terms of what we've done here, what we're talking about is a couple dozen. It's not hundreds. But that's because it's informed by what we call biological prior knowledge. Yeah.
Please.
Thank you very much, wonderful talk. One quick question is that when you do the PCA, the principal component, do you have a priori, you select those particular
Yeah, here, no, this was,
based on some bioinformatics or
no.
previous
Yeah, no, this was unbiased. There are other ways you can do it.
or unbiased?
Yeah. That's what we were surprised, actually, that it ended up in 2 different patient disease cohorts, but now there's reason to believe that that's actually real.
And this, how many patients?
This was like 17 or something.
17.
Yeah, small number, but obviously we're pursuing more.
So I had a quickie, Doug. So in this lovely story where one effectively anneals the various signal transduction forces that one detects and tries to determine whether the sum, it's not a simple sum, but the collective signal transduction data suggest or indicate the presence of a diseased cell versus a non-diseased cell. Can you correlate these signal transduction changes with pathological changes measured by whatever, antibody binding to cell surfaces, cell shape, morphology, invasive behavior, whatever?
Yeah, great question. We need a richer characterization of phenotype, basically. And this was pretty simple. The epithelial cells were dying or proliferating.
Yep.
Okay, we think with a richer characterization of what the cells are actually doing, we can actually make that a stronger model and then be able to measure maybe those as surrogates, if that's what you're suggesting.
Something like that.
Yeah.
Okay, thank you very much. Deeply appreciate it.